Yes. Thanks for the introduction. Hello, everybody. I was asked to talk about a national campaign we started a few years ago. Co-authors of this talk are a lot of people. I mentioned here Matthias Geiger, Björn Rulik, who are coordinating the GBAL data in our institute in Bonn. I want to focus on some difficulties we encountered. This might be interesting for some of you. I want to talk about the relevance of citizen scientists in such an enterprise. And I will show you just a few results, because the results are essentially what you expect to get from such a project. We have heard many examples during the past days. Instead, I want to spend a few minutes at the end of my talk to explain you a vision I have, how we will be integrating barcoding in a whole array of technologies that can be used for monitoring of species diversity. So, first of all, if you want to organize a national barcoding campaign, what you should do is to thank, in the beginning, Paul Hebert and the team here in Guelph. Because of the existence of the CCDB, because of BOLD, because of long lists of arguments compiled by the Canadians, which you can use to convince stakeholders and policymakers and funding agencies. So it's quite easy to start such a project nowadays. Of course, you need partners with laboratory experience, taxonomists, and especially you need partners who are motivated to invest time, who really want to work. I started to visit different natural history museums in Germany to explain what we would like to do, and I was happy to see that everybody was enthusiastic. They wanted to take part in such a national initiative. With one exception, the researchers in the natural history in Berlin told me, barcoding is not real science, we don't have time for such a thing. 3 years later, they called me, oh, can we join GBAL? They have seen the impact the project is having, and they wanted to be part of it, but it was too late because all the slots were already taken by other people. We also needed the support of universities. We found specialists for angiosperms, nematodes, fungi, and other taxa only in universities. We didn't have them in the natural history museums. So after some time, we had a nice network of institutes who wanted to take part, scattered over all Germany. And then you have to organize workshops, to sit down with them, to discuss behind closed doors how we want to cooperate. You have to agree on workflows for specimens, for data, for DNA samples, and so on. And the workflow is then quite complicated. But if you don't know how to do this, just visit Guelph and they will tell you. Then you have to agree on a division of labor, and you have to define some regional nodes. What we did was to divide Germany into 4 quadrants. Germany is not so big, you only need 4 quadrants. And then you identify institutes which will be responsible for sampling in their quadrant. We also divided the taxa among these institutes. This would mean that, for instance, our friends here in Stuttgart are responsible for the species list of Hymenoptera, and they are responsible to complete sampling of Hymenoptera. And other institutes who catch these animals should then send them to Stuttgart. All this didn't work perfectly. Some institutes collected only for themselves, others collected only a few taxa. But this is an important basis for cooperation to avoid discussions, misunderstandings, quarreling, and so on. Then it's important to know which other projects are already funded to avoid the duplication of efforts. So there are a number of projects in Germany. Michael Raupach, who is here, is organizing the barcoding of marine organisms. There's a large project in Bavaria. Axel Hausmann is here, is representing this initiative. They are barcoding everything they can get in Bavaria. So what we really want to know is what everybody's doing. We want to join forces, and we want to put all the data together. Then you need additional supporters because you want to collect in national parks, in nature reserves. So what we did was to visit the annual meeting of the officers responsible for these parks. We explained what we wanted to do, and it was astonishingly easy to convince them that it would be useful to collect within their parks, which normally you are never allowed to do. They were enthusiastic with the idea that in future we will be able to identify everything they have and to monitoring the development of these parks. This is what they really want to have, and they are not able to do it. We also needed the permission of the Federal Agency of Nature Conservation that we will barcode also red list species, which normally are absolutely forbidden to collect. Then having this basis, or the partners, the concept, we were ready to address the Federal Ministry for Education and Research because we wanted them to fund the project. So we organized a workshop with representatives of this ministry, and we agreed to write a white paper where we describe the whole project. This we did. We emphasized especially the utilitarian arguments because this ministry wants to support the German economy. They are not so much interested in species and nature. But there are enough arguments, also in combination with innovations that will be developed in such a project. So we wrote a white paper. It circulated in the ministry, and we got a very encouraging feedback. They told us, write an application and, well, let's say not more than 5 million euros. This was easy. We could have spent more, but okay, 5 million are a good sum. We submitted the application. In parallel, we all signed a memorandum of understanding, and then you have to be patient. You have to wait. We didn't have to wait long. This was surprising. Obviously, the ministry was prepared. They already had the money reserved for us. So the whole timetable looked like this. We needed nearly 5 years from the first idea to the start of the project. And most of this time was required for networking, for lobbying, for preparing the white book. And then things went relatively quickly because the ministry already knew what was coming. So we started in June 2012, and then immediately we started to travel again through Germany to visit natural history societies. We have several of these. These are sort of clubs that meet at regular intervals to discuss what they have been observing in the surrounding area. They collect beetles, they observe the distribution of plants, and so there are a lot of amateur taxonomists with excellent knowledge in these societies. So this, for instance, is a group that works in central Germany in the state of Thuringia. You must imagine for many decades after the Second World War, these people were under Soviet influence, and they had little resonance for what they were doing. And now suddenly they were asked to join a national initiative, and they suddenly realized that their knowledge was appreciated by a lot of people in our country. And this motivated them to participate. So currently we have 230 amateur taxonomists helping us. Many of them love beetles and butterflies. Nobody loves nemerteans. But we need all this support. We are recompensating these people for their efforts. We pay them 3 to 5 euros per specimen if we get the taxonomic name, if we get all the metadata, and if the specimen is valuable. We don't want to have 10,000 Drosophila melanogaster. We want to have valuable specimens. And this is not a reimbursement. It's impossible to reimburse these people. They travel the whole weekend through the country to find a single species. We cannot pay for that. But we want to show them that we acknowledge their efforts and that we appreciate this. Now, discussing the participation of taxonomists in such an enterprise, we've encountered a lot of opposition. And now I want to explain how we reacted, because maybe this can be useful for you. A frequent argument we heard, we don't want to support barcoding because barcoding is replacing our expertise. Now, what would we say? We are replacing only identification keys. And these keys are anyway often of little use because an ecologist will not be able to identify spiders using this book. You really have to be a specialist. So we are replacing this. And you have to explain them the difficulties ecologists have. They want to collect samples, they want to identify the material, they have to sort the material, they have to look for taxonomists, they don't find them, and when they deliver data, it's very slow. So the ecologists are really desperate when they try to cooperate with taxonomists. And if they find someone and they give this person a mass sample, you will hear, awful sample, it's really ugly, I don't want to work with it, it's boring. So, another argument is, you're saying we are responsible for the loss of taxonomic expertise. This is not true. This is some data on the specialization of professors of zoology in German universities. I compiled this some time ago. And you see there's a strong decline in the fields of morphology and systematics since the 1960s. And this went on till 1990, and since then this field of research did not recover. Others which gained are genetics, physiology, this is developmental biology, neurobiology, and so on. So we lost expertise in universities long before DNA barcoding was invented. What we really want to do is to reverse this trend. And it's important to discuss with taxonomists what they can do to reverse the trend. And we come up with some ideas, make taxonomic expertise more visible, write articles for newspapers, write articles for weekly journals. But this is not enough. You really should join ecologists. We have currently a large program in Germany funded by the German Science Foundation where ecologists try to find out the influence of diversity on ecosystem functions. And they, of course, want to know which species they have in the samples. But because of the experience they have, they change their subjects. They are working with guilds instead of species, or they work only with 5 pollinators because they are able to identify them. So taxonomists must help to improve the situation. They must help ecologists to get the names. They must make taxonomy more usable. And the best way to do that is to help to develop reference databases and to develop workflows for analysis of multiple samples. And soon we will be able to do that. For instance, tagging these samples to be able to mix them, use hybrid enrichment to fish out several markers from a single DNA soup. This will soon be routine, I hope. For many years, I have been observing that there's a Chinese wall separating ecology from taxonomy. We have to tear down this wall. And this not only does help the ecologists, it helps the taxonomists. Then I tell amateur taxonomists, you must be more than just someone who knows how to distinguish species. You know much more. Sorry, back. You know a lot about the biology of the species, where they live, their ecological niches, what they feed on, the adaptations, and so on. And this will not be replaced by barcodes. On the contrary, if ecologists are able to identify species, they will ask you, what do you know about the species? So your knowledge will become more important. Then we hear barcodes do not identify species. I hate this discussion because you have to start from the very basics. What is a species? We are running in circles. We are running in circles for decades discussing this issue, mainly because I think biologists are not trained in ontology. They are not trained in separating cleanly between the material reality and the consequence of using a language which requires a classification. So a species is a logical class. And therefore, there are so many discussions. So to make things easy, just tell people a species is what a taxonomist recognizes as a species. A consequence of this, a consequence is that our reference database is as good as the taxonomists who help to validate the reference database. So the taxonomists are responsible for the quality of this. And then it's nice to have examples like this one, a handful of musted flies identified by a specialist. We barcoded this material, and then we discovered some inconsistency here. Two closely related lineages with a single name. We sent the material back to the specialist, and he tells us, oh, sorry, there is obviously a cryptic species we overlooked. And here, a different case, one sequence with two names, and it turns out it's a case of misidentification. So in the dialogue between the experts and the barcoders, we get a much better reference database. And if this taxonomist dies, his knowledge will be conserved and will be usable by next generations. Then, yeah, sometimes we hear that we are killing jobs for environmental experts. These are people who earn money writing in environmental audits. But the contrary is the case. In future, they will be able to sign more contracts. They will be faster. They will be more efficient. They will earn more money. This is what we tell them. If it's true, I don't know. So we summarized all these arguments in a paper written in German language, and if someone is critical, he should first read this paper, and if afterwards there are still questions, we can continue the discussion. Okay, then we should train these amateurs. They don't want to do the wet lab work, but they want to know how to retrieve sequences from published databases. They want to know how to align sequences, and they want to know what the neighbor joining tree means. So we invite them from time to time to learn this, and they like it very much. And the consequences, we are starting to see a new type of publications. Amateurs are using barcodes, and they're using neighbor joining trees to show where a newly discovered species is placed on a tree, and this is nice. Now, important means of communication in this scattered community is a web portal, and this is not a duplication of Bold. We are using Bold, but we also have to have a portal in a native language because we are dealing with common folk. These are certainly not the citizen scientists, but this is the type of people we are dealing with, including members of parliament and officers in the ministries. Our webpage is in German, and it helps us to make our partners proud because their names appear here. They are specialists for certain taxa. And we use the page or the portal for progress reports. For instance, this is a list of snails. There are 43 species of hydrobids known from Germany. We only have 3 in our collection, so we need more collections. The map shows where specimens have been collected. And the green numbers tell you how many species and specimens already have been barcoded. So if you are a specialist for beetles, you want to help, you can enlarge the area around your hometown and see what has been collected. You click on a dot and you see which species have been found there. You can download this information and then decide what you want to do. We use this portal for the registration of experts, and then we learned from Guelph how useful it is to send them boxes with label tubes. The same numbers of the labels are already entered in a spreadsheet. People can download to enter the metadata and so on. So this whole workflow developed here in Guelph proved to be extremely useful for the communication with the collectors. A few statistics. We originally were interested only in specimens with full taxonomy. We collected in 3 years 22,000 species with full taxonomy. At the end of 3 years, we had 17,000 already processed, 71,000 barcodes. There are some official lists of the species occurring in Germany. The numbers are too small. Specialists know many more, but these are the lists published by the Federal Agency for Nature Conservation. And what we have after 3 years is 43% of the beetles, 61% of the butterflies, among others, thanks to the efforts of our colleagues in Munich, thanks to Axel Hausmann and his team. We have nearly 40% of the vertebrates, and you see where the weak parts are dipterans. During the past talks, we have met this problem again and again. There are many, many flies. They're very abundant, and there are few taxonomists. So we have to focus more on dipterans. Here, I think an institute failed. There's an institute responsible to barcode soil fauna, nematodes, collembolans, and so on. They didn't do enough, and therefore, for the next funding period, this institute will probably be excluded. So for the next 3 years, we hope to barcode another 14,000 species. The process is getting slower because now we have to collect those species that are less frequent. It's becoming more difficult. The geographic coverage has to be improved. These are beetles. Many have been collected in central and western Germany, in the north, nothing. Maybe we will organize a bioblitz to collect in this area. Then we are interested in the resolution of the barcodes. We check that from time to time, and it generally works nearly perfectly. These are some leaf beetles from central Germany. Each sequences can be assigned to a species in a perfect way. So we are happy with that. We are now able to identify difficult parasitic wasps, like here in the genus Tetrastichus, or in the pteromalids, which are a nightmare for taxonomists who work with this group. Now we can separate them, or here, hoverflies, Ferrophoria, 8 species, all look identical. An ecologist probably would give them the same name. We can distinguish them now. And then you have some dirt on a leaf. If you barcode this dirt, it turns out to be a certain rust fungus. There are, of course, a lot of byproducts, and I don't want to present them. Just mention that, of course, we discovered new species, new records, new host-parasite relationships, and so on. I want to talk about the vision for the future. Our planet is changing in a dramatic way. Not only the glaciers are disappearing, but also habitats at a large scale, and species are disappearing. And even in Europe, where most changes already occurred hundreds of years ago, we are seeing dramatic losses. For instance, 20 years ago, when you drove over a highway in summer, every few hours you had to stop to clean the windscreen. And you could buy special insect dissolving solutions in gas stations. And now you drive at the same time of the year, the same road, the windscreen will be clean. The insects disappeared. The abundance dropped dramatically. And the bad thing is we do not have data. If you ask people, they all remember that 20 years ago the situation was different, but we do not have data. There has been no monitoring program. And this is very bad because we cannot talk to politicians. We cannot convince them that something has to be done. So I think we should develop principles for automatization of biodiversity monitoring in the same way climate researchers are doing it. We are now working on a project we called an AMOT. We want to build an automated multi-sensor station for monitoring of species diversity. All the technology is available. We only have to put it together and to adapt it. Elements of this technology is automated sampling in combination with barcoding. So everything which is airborne can be collected automatically. We are building a malaise trap that can collect over 3 months without needing attention. So you leave it in the field for 3 months. Bottles are being filled one by one. You can program it. Maybe you only want to have on Mondays a sample over 3 months. This can be done. And then you bring the material to a lab to analyze the samples. We are developing this with university institutes, among others. This institute will develop a sampler for pollen and for airborne spores. Second technology is bioacoustic monitoring. There are many publications explaining how this can be done. The instruments that are being used are still very primitive. They have batteries and they have memory cards, and someone has to go there and change the memory cards. In future, we would like to transmit the information to a receiver station that is far away, and we would like to analyze these signals automatically. This means you need a sound archive. Maybe Bolt would be a good place to build up such a sound archive, and you need good software. And most of the work that has to be done in the moment is to improve the software to filter out from the soundscapes those signals you're interested in. And currently, we have several institutes with computer scientists who try to improve this type of software. The third technology, automated image recognition, you can imagine why there has been a lot of research in this field. We can use the same algorithms to identify species. However, until now, the camera traps are still very primitive too. They need batteries, they have memory cards, and then you have manually to go through the images. This can be done much better. There's a prototype of a camera currently running in a forest in Cameroon. This camera sends signal to a receiver station, and there you can observe which apes or monkeys have been crossing this path, and the system tells you which species has been identified. And this is being recorded, so you look up the database and you see, okay, yesterday around 10 o'clock, a female chimp called Lucy crossed the path. This is possible. However, we need a better resolution, especially if you're dealing with things like closely related moths. And again, there are several institutes currently trying to improve this. And then I discovered artificial noses. There's an institute in Dortmund developing a system that detects tiny amounts of molecules in air. They're using this in clinics. There's a patient exhaling into this machine, and the machine detects if this person has tuberculosis or not, because tuberculosis bacteria produce chemicals that can be found in the air. And we are now testing if this method can be used to detect the scent of flowers or the smell of fungi. It's essentially based on gas chromatography, and then there's an ionization chamber. The molecules are detected here on a Faraday plate, and it's very sensitive. This is the institute doing this. So this is our vision for the future. We'll have scattered over the continent many automated observation stations. We will have central stations where the data are being processed, and then we will be able to do the same thing climate researchers are doing. We are able to model, but we need better data to model scenarios. But I think if a few years from now, this will be possible. Thank you. Yeah.